So here we are in the bird collection in the Florida Museum of Natural History, and we have Andy Conrader, who is the bird collection manager, to talk a bit about the collection.
We have about 55,000 specimens of birds in our collection, and it's kind of a half mix of skeletal skeletons of birds and skins of birds. And it's one of the strength of the collection is a skeletal collection. It's one of the largest in the world in terms of both diversity and total quantity of skeletons from all over the world. We have about half of the species of birds in the world represented in our skeletal collection. One of the main uses, of course, of the bones like this, and the reason why we want to disarticulate is to identify fossils. And that's the main thing that the curator of the collection, Dave Stedman, studies. And he'll go into archaeological sites that are maybe 5,000 years or 10,000 years old and find the distal end of a tibia tarsus, maybe this little bit of a tibia tarsus, and have to identify it. And so he'll bring out a whole bunch of different, he'll know just by looking at it that it's a heron. He'll probably know just by looking at it it's a spoonbill because he's such an expert at it. But the way that he would identify it would be to compare it with extant species. And if it differs slightly or differs majorly, then it might be an extinct species of spoonbill.
So ornithologists tend to take a certain set of standard measurements when they're studying the morphology of birds. One of the common ones is what's known as the wing chord. And it's called the wing chord because on the live bird, the wing actually arcs a bit. So there's a curvature to the wing. So you're measuring the chord of the wing. And you can see here, this is a specialized ruler. It's got a butt end to it. It fits right up nicely here against the bend of the wing. That's the carpal joint of the wing. And you can see it extends down to the tip like that. Of course, a toucan, probably the most fun measurement, is the bill measurement, right? And this, I'm not going to touch the bill because I don't want to damage it right now, but with these metal calipers, but you can see here you can get a nice measurement of the length of the bill. You can also get a measurement of the depth of the bill and also the width of the bill. These are all very useful measurements because for one thing we know, biologists who study species of birds that eat fruit, they know now that the size of the bill is very much correlated with the size of the seeds or fruit that the birds eat. And birds are important dispersers of seeds. It's an important ecosystem service that they provide. And so the shape of the bill provides a lot of information about that ecosystem service that this species can provide.
As you can see here, we have, over the 100 or so years that this collection has been growing, we've accumulated quite a few specimens. So if you're looking for changes over time, you could use this collection. If you were looking at, you might wonder why we have so many, but cardinals are going to differ from different times of the year. They're going to differ by male and female. They're going to differ by different age, and they might differ subtly geographically. So you want good sample sizes of each one. If you're looking at something like molt, where they're changing their feathers, they only molt their feathers probably from July into September. So you need good samples from that, and they might molt earlier up in North Florida or in South Florida than they do in North Florida. So just to look at these things, you need nice sample sizes. It'd be nice to have sample sizes this big of all the species, but of course that would not be possible right now. Like I said, we get almost all of our birds, recent birds, are from wildlife rehabilitation clinics all over Florida.
One of the interesting findings that has come out of our research in Peru has a lot to do with the type of light the birds use. And one of the things we did in the field is we actually measured the eye, the size of the eye, and it's actually simpler than you might think. All you do, we found, if you just take a measurement of the width of the eye, like this, it's highly correlated to actual eye measurements from preserved eyes, people preserved in alcohol. And so we can use those field measurements of eyes to actually create a morphological index of how sensitive to light birds are. And we find that the birds that tend to disappear from very brightly lit agricultural habitats tend to have larger eyes. And and we hypothesize it has to do with just so much brightness interfering with their ability to persist in those types of really bright landscapes.